Hello everyone, welcome to SG Pharma Trainings. We are here for a demo class on pharmaceutical microbiology and importance of microbiology in pharmaceutical industry. Myself, Bhavana Tatineni, completed my PG in MSc Microbiology and I am having 18 plus years of experience in quality control microbiology in various pharmaceutical companies. I will be with you till completion of this course. Before entering into the session, I will explain why microbiology is so important in pharmaceutical industry and how you can learn each and everything about it. Those who are interested in microbiology, they can learn each and everything in this course and it will be definitely beneficial for all those candidates who are looking forward and to make a career in microbiology. Let's start the demo. There are all the syllabus we covered in this course. First, introduction to pharmaceutical microbiology. Microbiology is a branch of biology dealing with study of microorganisms. Organisms that are usually too small to be seen clearly with unaided eye. These organisms include bacteria, fungi, viruses, protozoa, and algae. Next, we'll go flow of microbiology laboratory and facility design. The microbiology facility should be designed in such a way that it should avoid the cross contamination from person to sample or from sample to person. And the flow of materials and the movement of persons will be discussed in this chapter. Next, media receipt, storage, and preparation. Media is a growth media required for microbial growth. We will procure the dehydrated media from the manufacturer and will store at room temperature. Some media require refrigeration. We will store accordingly and will prepare the media as per the manufacturer instructions by adding the how much water required, as how much media should be weighed. Everything will follow the manufacturer instructions. After preparation of the media, we'll sterilize the media and we have to qualify the media before usage. What is media growth promotion test? It is a quality test to check the whether media promoting the growth or not. We will inoculate the known number of microorganisms and we will check the recovery. Each and every prepared lot should undergo growth promotion test. Next, culture handling. Cultures are the standards which we use in the microbiology laboratory. These are the controls we will perform along with sample analysis. Whether the culture grown during challenging, it means the test which we performed was correct. Next, types of water systems in the pharma industry. How the water system will be validated. Different types of waters will be used in the pharmaceutical industry. These are raw water, purified water, water for injection, and pure steam. How these are all will be generated, we will discuss in this chapter. From raw water to pure steam, different purification steps will be involved. And whenever the new water system was constructed, we have to validate the system. What are all the steps involved in the validation? We will discuss in this chapter. Next, water sampling, testing, and trending of the data. Microbiologists should collect the samples as per the schedules. What are the precautions to be taken while sampling? How the testing will be conducted by the microbiologist? How data will be recorded? How the results will be recorded? How the interpretation of results will be recorded? We'll discuss in this chapter. Next, trending. After obtaining the specific data, we'll prepare the trends. These are monthly trends, annual trends. As per the requirement, we'll define. Next, types of environmental monitoring and data trending. Environmental monitoring is collection of air samples in the clean rooms. We have to check the air quality. This is by two methods: settle plate monitoring and air sampling methods. For these also, we'll prepare the trending. Alert and action levels will be calculated based on the trending data. By seeing the trend data, we can come to know that whether the consistency is maintaining in the facility or not. We'll check either it may be for water or for environmental data. Next chapter was how the selection of locations for environmental monitoring will be done. We'll expose the plates at a different location. How the selection of location will be selected? Where there is more chances of getting contamination, those sampling locations should be chosen and the risk assessment should be performed for each and every location. And how many number of locations should be selected also will cover in the risk assessment. Next, microbial limit test. This limit test will check for water samples, API samples, finished samples. Whether the sample meeting the predefined specifications for total aerobic microbial count, total combined yeast and mold count, and test for specified microorganisms. These are E. coli, Salmonella, Staph aureus, Pseudomonas, Enterobacteria, Clostridia, and Candida. Next, what is bacterial endotoxin test? By this test, we can come to know how much endotoxin content present in the sample. This is also for water samples, API samples, and finished samples. What is sterility testing? In this testing, we will check the absence of microorganisms. These are for sterile products. Sterile products should be free from all living organisms. By sterility testing, we'll check the any contamination is there in the sample or not. Next, bioassays of antibiotics and vitamins. Bioassays will be used to calculate the potency of the drug. This is applicable for antibiotic and vitamin assays. Two types of bioassays will be performed: couplet method and turbidometric method. In this chapter, we will discuss in detail. Next, preservative efficacy testing. This is also called as antimicrobial effectiveness testing. We will check the preservative which we added in the drug product. It is working throughout its life cycle or not, its shelf life or not. We will check by preservative efficacy testing. For stability samples, we will check the preservative efficacy testing. Next, analytical method validation in microbiology. Each and every method which we used in the analysis of microbiology samples should be validated. By the method validation, we can come to know the sample testing which we followed correct or not. In this also, we'll challenge the known number of microorganisms and we'll check the recovery. Next, types of equipments, instruments, and its uses in the microbiology laboratory. We'll use different types of instruments and equipments in the lab, like autoclaves, incubators, biosafety cabinets, laminar airflows, air samplers, microscopes, etc. Each and every instrument uses and its principle we'll discuss in this section. Sterilization techniques and validation of autoclave. What is sterilization? Sterilization means removal of all living organisms. There are different types of sterilization techniques. Those are chemical sterilizations, heat sterilizations, radiation, and filtration. Generally used methods are heat sterilization and filtration in the pharmaceutical industry. In the heat sterilization, we will use moist heat, example autoclave, dry heat, example ovens, hot air ovens. And for filtration, in this filtration, we will use 0.2 micron or 0.45 micron filters for removal of microorganisms. This is used in the filtration of liquid samples. Next, handling of biological indicators. Biological indicators are the spore indicators which we challenge in the autoclave and dry heat sterilizers. We'll add 10 power 6 cells of biological indicators in the autoclave cycle. We'll check the recovery. The recovery should be zero. There should not be any growth. How the biological indicators qualified after receipt, we'll discuss in this chapter. Next, microbiological monitoring of gases. Nitrogen and compressed air will be used in the pharmaceutical industry. These will come directly in contact with the product. So the quality of nitrogen and compressed air will be monitored by total aerobic microbial count, and for nitrogen, total anaerobic microbial count. And next, disinfectant validation. What is disinfectant? The chemical substance which we use for sanitation of surfaces, that is called disinfectant. So different types of disinfectant will be used in the pharmaceutical industry. Bactericidal, fungicidal, virucidal, sporicidal. These are all the disinfectant different types, and each and every disinfectant which we used in the laboratory should be validated for its concentration and for its contact time. In these two methods, use dilution and surface challenge studies will be conducted. Next, sterile manufacturing facility and media fields. Sterile manufacturing facility means the aseptic processing area which is used for manufacturing of sterile products. Media field, also known as process simulation study, which is carried out to assess the capability of aseptic process. In this chapter, we'll discuss what is the frequency of media fields, what if media field failure happens, all this we'll discuss. Next, handling of OS in the microbiology. OS is out of specification. If our sample crosses our predefined specification, then we have to initiate OS investigation. If microbiology samples got OS, what precautions to be taken to avoid the reoccurrence, how the OS will be handled, we will discuss in this chapter. Next, cleaning validation and handling of clean room bioburden samples. Cleaning validation is validating our cleaning method which is used in the manufacturing facility for all instrument surfaces. After cleaning, we'll collect the bioburden samples and we'll check the cleaning efficacy. This is by two methods, swab samples and rinse samples. In detail, we'll discuss later. Next, handling and destruction of biological waste. We are growing the microorganisms. We are handling the culture media. We are generating so much amount of biological waste in the microbiology laboratory. As you all know, biological waste is very harmful for our environment. How this biological waste can be destructed, how we'll handle to avoid cross-contamination and to avoid spreading of infections. This is very critical part. We'll discuss later. Next, identification of microorganisms. If any unknown microorganism we found in our sample, we have to check whether which genus level or which species level the microorganisms will belongs to. By that, we can come to know the source of contamination. It is useful in the investigation to avoid the reoccurrence of the cross contaminations or OS results. Next, biopharmaceuticals. Microbial or microbial byproducts which are used in the therapeutic use will be manufactured in biopharmaceuticals. How the biopharmaceuticals will be manufactured, we will discuss in this section. And example for biopharmaceuticals are vaccines. Next, microbiology best laboratory practices. A microbiologist should know the practices to minimize the contaminations and to avoid the cross contamination of samples, to mix up of the cultures, everything he should be trained. And a microbiologist should capable of generating the accurate data, how to interpret the results, how the precautions should be taken while handling of the cultures, everything will be covered in this section. Next, guidelines. What guidelines suggesting for microbial testing? What USP saying? What EP saying? What WHO, ICH guidelines? What are the chapter numbers? What are the limits mentioned in the guidelines? We will discuss in this chapter. Next, data integrity and data mapping. Data integrity from generation of data to destruction of the data, this called data life cycle. All data are complete, consistent and accurate throughout the life cycle. The data might be electronic data, paper based or both hybrid. So all the principles, all Alcoa principles will be discussed in this section. Next, audit overview, types, steps and audit readiness. How we have to present our laboratory during audits, how we have to behave with auditors, how our lab should be readiness each and every time for audits, we'll discuss in this section. Believe me guys, this course, entire course will be very beneficial to each and everyone. Thank you.